All right, it's my pleasure to introduce Amber Johnston. She's a second-year PhD student at the University of Maine, working with Dr. Debbie Bouchard and Dr. Mark Polinsky. Her research focuses on furthering our understanding of the host-pathogen relationship between infectious salmon anemia virus and Atlantic salmon. Thanks so much for being here.
Yeah, thank you for having me. I just want to get started by acknowledging my co-authors for all their support on this work. Dr. Roundsville for all of his molecular expertise, and my co-advisors, Debbie Bouchard and Mark Polinsky, doctor in front of all three of those names. So, all right, without further ado, one of the most infamous diseases affecting the Atlantic salmon industry is infectious salmon anemia caused by infectious salmon anemia virus. First described in 1988 and reported in the US in 2001, the virus not only causes severe clinical disease, but is also one of the most intensively managed finfish pathogens globally due to its capacity to create epizootics. ICV is an enveloped negative-sense single-stranded RNA virus in the family Orthomyxoviridae, and if that sounds familiar, it's probably because Orthomyxoviridae is also home to influenza viruses. With that, the ICV and influenza virus virions are very similar structurally, and all viruses in this family have segmented genomes. ICV in particular has a genome consisting of 8 segments that code for at least 10 proteins, and segments range from about 800 to 2200 base pairs in length, with the whole genome coming in between 13 and 14 kb. Similar to other viruses in the family Orthomyxoviridae, ICV is highly genetically diverse. One of the first ways you can group ICV isolates is by geographic origin. So the photos I'm showing you here are phylogenetic trees generated from segment 1 and segment 4 sequences from about 40 different ICV isolates, and you can immediately appreciate the separation here with isolates clearly falling into 2 distinct clades, one marking isolates of European origin, known as the EU genotype, and one marking isolates of North American origin, known as the NA genotype. It's important to note here that while in Europe there have only been reports of EU genotypes, in North America we have and continue to detect both, meaning that any genetic studies focusing on ICV in this region must encompass both genotypes. Within these 2 genotypes, ICV isolates can be further divided based primarily by what goes on in segment 6 of the genome. And this segment is unique because of the highly polymorphic region, or HPR, found toward the 3' end. The figure I have here is a snapshot of the HPR of about 50 different ICV isolates, and with this, ICV is broken up into 2 predominant categories, one of which is widely considered to be the wild type, has a fully intact segment 6 HPR, and as a result does not cause clinical disease. Isolates under this umbrella are commonly known as HPR0 isolates. The other group of isolates contain a deletion event in the HPR, and these isolates are known as HPR delete isolates and are associated with the causation of disease. While the exact relationship between HPR0 and HPR delete isolates remains largely unknown, it's hypothesized that HPR delete isolates are the result of a deletion event occurring in an HPR0 isolate. And the unknowns surrounding this potential transition are super important because while detections of HPR0 or delete often require some degree of management action, given we have no knowledge on why, if, how, or even if this event even happens, it's challenging to manage an ICV HPR0 detection effectively. Think of it as like having a firecracker in your hand and not really being sure when or if it will ever go off. So there are a couple things inhibiting how we can study this phenomenon. While we're able to grow boatloads of HPR delete under in vitro culture conditions, we are not afforded that same luxury with HPR0, meaning that it's challenging to do any in vitro or in vivo studies with ICV HPR0 without an HPR0 positive fish. So say we have an HPR0 positive fish, even if the tissue carries a high virus load, which is very rare for an HPR0 infection, when you go to sequence, finding that small bit of viral RNA in host tissue is essentially like finding a needle in a haystack. And while this phenomenon isn't ICV specific, it is an important problem to address for ICV, as to date there are no published methods that effectively capture the genetic diversity of the virus that are optimized for both cultured cells and sequencing directly from tissue. So with all of that background and context in mind, the overarching goal of this study is to explore genetic variation of nearly 30 years of archived ICV isolates collected on the northeastern Atlantic seaboard. While there's been a lot of work done on comparing ICV genome segments individually, there are few publicly available whole genome sequences of ICV, especially in this region. So with this project, we hope to collect hundreds of isolates from our laboratory and industry partners and generate that missing genome data. And by taking this approach, we'll be able to compare whole genome diversity, identify any regional patterns, including things like source tracking and other epidemiological dynamics. And lastly, we hope to use all of this generated sequence data to maybe even explore that cryptic dynamic between IACV HPR0 and HPR delete. Before we do any of that fun stuff, though, we need a consistent, robust sequencing method. As like I mentioned previously, there are no published methods that, one, can encompass the full genetic diversity of ISAV isolates, and two, are optimized for both cultured cells and tissues. Which brings me to the specific goal of today's talk, which is to develop that all-encompassing ISAV sequencing method. So full disclosure, I'm not all the way through this stage yet, but I'm pretty close, so I'm excited to be here to share what I have so far. All right, so what do we need this method to do? Well, we needed this method to capture NA and EU genotypes, capture HPR0 and HPR delete isolates indiscriminately, and lastly, at some point in this process, we needed to increase the amount of viral genetic material relative to hosts. So with that last point in mind, the first step of our method, and what I'll be focusing on mostly today, is the preamplification process targeted at increasing the amount of viral nucleic acid prior to nanopore sequencing. And we chose nanopore here in an effort to better capture any ISCV isolate co-infections with longer read lengths. So the first step of this is to design virus-specific primers. And there are two approaches to this. The first and most straightforward is to design primer pairs that grab specific whole genome segments. This method has been used, for example, with influenza virus sequencing. Next is to create more primer sets that amplify partial segment fragments. So this alternative route has been published for use with other human pathogens like Zika virus, and a publication using this for Piscine orthoreovirus is currently in press. So as I go through this, and given this is a method development focused talk, I'll be telling this story as it happened, focusing on the methods and results of each step of the process before moving on to the next. So for both methods, we used a high-fidelity cDNA synthesis kit with random primers prior to undergoing virus-specific preamplification. And first focusing on the whole segment preamp approach, which required the least number of primers and objectively made the most sense to start with, using Genius Prime, I downloaded and aligned publicly available sequences from NCBI GenBank, where I then identified primer candidates and regions of consensus at the ends of each RNA segment that, when amplified, should target the whole segment. Following initial design, primer pairs were screened for quality using IDT's oligoanalyzer tool, and then specificity using NCBI Primer Blast. In total here, I designed 10 primer pairs to test on the 8 segments, each creating products that range from about 800 to 2200 base pairs in length. I tested these 8 pairs individually against purified IACV North American HPR delete and IACV negative salmon tissue, subjected them to preamplification PCR with those primers, and visualize the amplified products using an eGel system. All right, as you can see here, I unfortunately had no success. So this little glimmer of hope that you see right here was not repeatable, as you can see also indicated with this little black box here. We have some thoughts on why this may not have worked, but I'll touch on that more toward the end. All right, so while it would have been nice if that would have worked, after little to no success with the first 4 segments that we tried, we adapted and redirected. So moving into the partial segment approach, I similarly created my alignments, and using the consensus sequence as a guide, I created 2 to 4 primer pairs per segment that tiled their way through the whole sequence. The goal being to pool the primers that capture fragments A and C, pool the primers that capture fragments B and D, run those amplification reactions separately, and pool the whole amplified genome before library preparation. Similarly, after creating these primers, they were all screened for quality and specificity using IDT oligoanalyzer and NCBI Primer Blast. Overall, this process yielded about 23 different primer pairs that ranged in amplicon lengths of 350 to 900 base pairs. I tested these primers on North American HPR delete, HPR0, EU HPR delete, and IACV negative tissue. We similarly amplified each of those cDNA samples with each primer pair individually, and that product was then analyzed using a fragment analyzer. So before I share exactly how many of these pairs worked and how many did not, I want to explain how we measured primer success. So the photo I'm showing you here is the output of our fragment analyzer, where you can see fragment size, or in this case, amplicon size, along the x-axis, and fluorescence intensity along the y. High fluorescence equals high amounts of fragment in that specific size range. So these four panels here are an example of a primer pair that worked well, capturing all three of the IACV genotypes or variants that we screened, not reacting significantly with host, and showing a high quantity of product within our expected fragment size range, highlighted here by the boxes. And I just want to note that there's about a 20-fold difference between this y-axis and this y-axis, so these little peaks here are roughly equivalent to these little peaks here. All right, so this is what a bad pair looks like, only producing amplicons of target size in one or two of the variants. And the good news is that overall, when we initially screened all 23 of these primer pairs and analyzed the results, we found that 20 out of 23 of the pairs worked as expected and 3 did not, capturing over 90% of the viral genome. But as you all know, close only counts in horseshoes and hand grenades. So as soon as I got back from the Western a couple weeks ago, I revisited this. And when I modified things like the annealing temperature and redesigned some of these primer pairs that were giving me issues, we did ultimately have success with all 23 of the pairs. So that was really good news. I feel very blessed by the science gods with this one. So we took that and ran with it. Encouraged by that success, we moved on with the next step of testing how these oligos behaved when they were all pulled together. 11 and 12 primer pairs is a boatload of primer to put into one pool, so we just wanted to make sure that things were working as they should. We tested this at 3 concentrations, 500, 250, and 100 nanomolar per primer, and we screened each pool at each concentration on the same samples we used in the previous preamp experiments. And amplified products were again analyzed using a fragment analyzer. So what I'm showing you here now is a fragment analyzer generated gel image of the amplified products in each primer pool for NA-HPR delete, HPR0, UHPR delete, negative tissue, and water. So the first panel here was amplified with 500 nanomolar of each primer in the pool. The shaded area in green is our expected virus amplicon size range, and any products below the dotted line are likely primer dimer, which we anticipate being removed during library preparation. So you can definitely appreciate some noise here, but 500 nanomolar of 12 different primer pairs is a whole ton of primers, so we weren't really surprised by this initially. What was exciting, though, is as we decreased primer concentration, we saw a considerable reduction in a myriad of things. First, we saw a reduction in host-associated non-target amplification, indicated by the lack of banding in our controls being phased out as we decreased concentration. We also saw a reduction in potential primer dimerization, indicated by the phasing out of the highlighted bands here. And lastly, we saw a reduction in apparent virus-associated off-target amplification, indicated by the phasing out of some of the smaller bands that we were seeing here. They're super faint, but they're not here anymore. So all of this done while retaining the amplicons that we were capturing in our expected size range. So looking at overall conclusions here, while we didn't have success with whole segment amplification, after some troubleshooting, using the partial segment approach allowed us to capture the virus genome in both genotypes and both ICV variants in totality. So there are a handful of reasons why we think we may have had more success with the short fragment approach, mainly being that large fragments of RNA are relatively difficult to preserve and regardless of storage conditions. And when it comes to actually deploying this method, we could be working with tissues or extracts that have been preserved for decades prior to whole genome sequencing analysis. So while it would have been nice to have less primers to work with here overall, by shortening our target fragment length, we're able to capture viral RNA from even some slightly degraded samples. Lastly, while there's still more work to go on determining the exact dynamics of our primer pairs and pools, initial results show little to no cross-reactivity with host genetic material, minimal dimerization, and only mild off-target reactivity. Goal here is to deploy this method on samples collected all over the northeastern Atlantic seaboard, developing this easy-to-use, consistent, and robust sequencing method to catch the genetic diversity of IACV is the first step in that process, and things are looking pretty good so far. So I'm running a library and putting it on the flow cell tomorrow, so we'll see what happens. With that, I want to acknowledge the University of Maine Cooperative Extension Tick Laboratory, specifically the lab manager, Ann Bryant, for getting me reagents when it was challenging to purchase said reagents, as well as the NACA between the University of Maine and the USDA ARS for funding me being up here. So, yeah. Thank you.
Awesome. As always, great presentation, Amber. Thank you. Exciting work. Folks, if you have questions, feel free to unmute yourself or put them in the chat. Anything out there? Alrighty, Amber, thank you very much.
Yeah, thank you.